So as scientists, we often hope that facts alone will change people's beliefs, and many times they simply don't. So people tend to stick to what they already think, and new evidence doesn't always sway them. A classical way to think about this is Pavlovian learning. So at first, a thirsty animal hears a tone that means nothing, and whereas an actual juice reward elicits a large burst of firing in its dopamine neurons. This occurs via a largely hardwired synaptic glutamate excitation. Now, over repeated trials, the animal's brain builds a predictive model using the tone to predict the reward with the goal of sending an equal and opposite GABA prediction. So eventually, the reward itself no longer causes a burst because it's fully predicted. And if the expected reward fails to appear, the unopposed prediction produces a pause. So this pause is very interesting because it represents a contradiction between the animal's belief that the tone will bring juice and the fact that it simply didn't. So how does the brain decide when to give up a belief? According to dogma, stubbornness is the default, and pauses are essential in overcoming stubbornness. Multiple optogenetic studies have artificially created or blocked these pauses supporting the dogma. Yet I'd argue that the synchronous patterns produced by optogenetics are a better model of the synchronous pauses seen following a surprising air puff to the face. By contrast, the pauses that you see when an internal belief is challenged are quite different. They're learned, they're not hardwired, their timing is irregular, and it also occurs in only a sparse subset of cells. Okay, now by contrast to this, again, the optogenetic perturbations are global and synchronous, and those mimic a hardwired aversive sort of signal that is highly synchronous and essentially involves all dopamine neurons. So other approaches have been used. So one is altering the GABA release upstream. However, we know that there are collaterals that can affect many other cell types. Finally, another approach is removing the GABA A receptor specifically in dopamine neurons via genetic knockout. However, genetic knockout leads to compensations, and light-gated GABA A receptors are essentially the same as optogenetics. And finally, traditional pharmacology lacks cell specificity. So there's a lot of issues here. None of the methods offer a clean, precise way to block only the natural GABA-driven pauses in dopamine cells without other confounding effects. So my lab developed DART, drug acutely restricted by tethering. This allows us to block native receptors on specific cells in an acute, controlled way. And this might let us isolate the effect of GABA-A-mediated pauses in dopamine neurons alone. Here's the basic idea. So we use an AAV to put a halotype protein HTP on dopamine neurons. That's this red protein that's going to show up here. And later we introduced gabazine DART, a small chemical that tethers specifically to HTP. It creates a high local concentration of the drug only on the surface of dopamine neurons, and this can block the native GABA A receptors. It blocks only the natural synaptic input to these neurons and spares everything else. When we do this, we mix in a small amount of this tracer called Alexa 647 DART. This allows us to measure how much ligand we actually delivered. The tethered compound stays stable for a couple of days, and so it allows us to do histology in each animal. And you can see this is a quantitative measure of how much chemical was tethered. And in control animals, we use a virus that's simply unable to bind the ligand, and this accounts for any effects of the AAV or the ambient ligand. In slice experiments, ambient gabazine DART is inert, whereas tethered gabazine DART potently blocks GABA-A-mediated synaptic inhibition. And you can see that right here. Now, we then moved on to an in vivo experiment, we recorded single dopamine units while puffing in gabazine DART. And in controls, you could see that nothing happened, and this tells us that the ambient ligand is not doing anything. Now, in animals with the active construct, we have observed a few things. The first thing was that the firing rate of the dopamine neurons stayed about the same. This surprised us initially, but looking at the literature, there is a literature suggesting that the pacemaker activity of dopamine neurons is under very tight and rapid homeostatic control. We also found that the bursts were unaffected as we had predicted. And finally, when we looked at how often there was a missing spike, in other words, a pause, we saw that this dropped markedly. In other words, in the controls, we'd see these periodic missing spikes, and that simply just didn't happen after gabazine DART, okay? So next, we did this Pavlovian task where mice hear a tone. They're thirsty mice, and they get this sweet water reward. Eventually, after several days of this, they start to anticipate it, and you can see that by this anticipatory looking during the tone. We normalized to their day 10 levels of anticipatory licking, and on day 11, we infused gabazine dart, and then suddenly omit the reward after the tone to see how quickly they extinguish. In other words, how quickly they can change their belief that the tone predicts the reward. Now, in the gabazine dart manipulated animals, our hypothesis is that if you block the dopamine pause, the animals will be stuck in this default mode of stubbornness. Our actual result was very, very different. We found that extinction got a lot faster, much faster. We did many controls in these experiments. For instance, we included a second tone, tone B, and that basically they didn't respond to at baseline. And this allowed us to interleave conditioning trials with the extinction trials. And these are done sort of on random interleave trials. And this allowed us to both keep the mice engaged in the task by having them know that there are rewards to be obtained. And it also allowed us to independently measure extinction and conditioning in every animal. Looking at individual variability, we found that the mice that learned the fastest in this new tone happened to also be the mice that were the most perseverative. Okay, so we actually saw this positive correlation. When we looked at the gabazine dart animals, we found that this relationship was eliminated. In other words, we eliminated this perseverative phenotype without affecting the ability to condition to new tones, new key reward associations. All right, so we next measured dopamine in more detail using fiber photometry. We used GrabDA3 in the lateral shell of the nucleus accumbens. Sure enough, in the control mice, we saw that when a reward was omitted, there was this large pause in the dopamine. In the gabazine dart mice, the pause was basically blocked. And we looked at many other things like bursts to a new tone, and everything else looked normal. And that basically matches our single unit recordings, which showed that gabazine dart is an incredibly precise manipulation of only the synaptically evoked pauses. Looking at individual trials in individual mice, we actually found this really great signal to noise. And these omissions here I'm encoding as a yellow, indicating it's -1, the deepest pause during the probe trials in any given animal. And we found that in individual mice, these pauses were always very strong right at the beginning of the process. And in some animals like this one, the pause didn't seem to go away. It seemed to just keep happening. And you can see that here in this smooth average where -1 is the starting value of the pause, and it very, very slowly goes away down to 0. And this happens to be an animal that was very stubborn, very perseverative. It just kept licking, and it didn't stop licking. Okay, so we see that in this animal, there's a lot of perseveration, and that goes along with this persistent pause. Here, I'm showing the average over day 11, this day here. Now, in other animals, they were less behaviorally persistent, and in those animals, their dopamine pauses also seemed to persist for a shorter time. And finally, in animals that received gabazine darts, where we forced the pauses to be gone from the beginning, we saw that basically behaviorally, they extinguished very fast. Okay, so basically we see this really clear correlation between the behavioral perseveration and the persistence of the dopamine pause itself. And gabazine dart seems to shift us along this natural relationship. But in fact, there are some natural animals that rapidly eliminate their pauses and also extinguish fast together. The final thing we did was we looked at the joint dynamics. And so specifically in this example animal, we see that the pauses seem to go away on their own naturally before the animal extinguished. And again, this is an animal that did not get gabazine dart. In other animals where this process happened faster, it seemed as if the pauses almost went away even faster before it dropped down. Kind of as if the pause has to go away first before the behavior changes. And finally, to show that this is a causal relationship, the gabazine dart manipulation basically forces the animal to eliminate pauses. And in those animals, every single one of them extinguishes fast. All right. So what does this tell us? Basically, it shows us that the pauses are naturally going away first before animals can extinguish. And we could accelerate this process by forcing the pauses to first go away and then extinguish. The only explanation that makes sense in this data is that these natural pauses are basically preventing extinction learning. They're basically there to solidify this internal belief. So that's very interesting. It tells us that the default is not stubbornness. In fact, the default seems to be rapid extinction. And to get stubbornness, you need to have this large dopamine pause to protect the belief. Okay, so that's really interesting. Now, again, there are many studies that seem to contradict this. However, I think all of us can sort of look at this and acknowledge that the other manipulations either didn't have cell specificity or had other circuit effects, or when they were cell-specific to dopamine neurons, they were studying a different thing. They were studying these global synchronous events that probably do encode the learning of new aversive things, but they don't necessarily update an internal belief. Okay, so the take-home points are that first, the connectome controls neural spikes in closed loop, and it does so in ways that we don't fully understand. With new tools, we can have more precise manipulations of the connectome itself, and with new tools, we gain new insights and ideas. And so this has been a very surprising result. It's been, kind of really pushed the boundaries of what I expected, and we've had to just sort of stay the course and follow the data. And I hope that you kind of consider this data as potentially changing the way that you think about dopamine pauses. And with that, I want to thank my lab, particularly Sasha and Renee and Haidun, who collected the data that I showed today. And if it's possible for me to take questions, I will. And if not, please feel free to reach out to me by email, whether you want to talk about this data or if you'd like to get reagents to collaborate. Thank you.